Alright, let's take a look at enzymes, specifically this catalase enzyme that we did our experiment on. If you've seen the protein YouTube video, then you'll know that what we're looking at here is a protein. Please remember that we had some technical issues on the last YouTube video. Hopefully we won't have them here on this YouTube video. Now here's this alpha helix kind of structure that we talked about. Remember, that's a secondary structure. That would be defined. It's a secondary structure that I'm talking about because I'm talking about the structure from here to here, right? So that part right there is a secondary structure. Just as the part from here to here would be another secondary structure, here to here is a secondary structure. But together, all of it together would be a tertiary structure because, again, we're talking about a bunch of secondary structures making one polypeptide. Now that one polypeptide has a bunch of different little parts to it. As I said, this alpha helix, this random coil here, this piece here that's a kind of a beta-pleated sheet. Each piece is defining an area that is chemically different and creates a space that maybe here it's hydrophobic, here it's hydrophilic, here it's ionically charged in a negative fashion, over here it's positively charged. And when it creates an environment that's alluring to a specific molecule that would fit in this place, fit in this spot here, and would be chemically attracted to be positioned in order to increase the likelihood of a chemical reaction happening. Now, let's take a look at this enzyme here. This is the enzyme in pink. This is the word that we call a substrate. And a substrate is the material that's going to be reacted, the reactants, if you will. The substrate could be one substrate, or there might be two, depending on the chemical reaction and what the product is. In this case, we're going to take a maltose, which is going to be the substrate, and we're going to break it up into two glucoses. This has to happen in your body. If you eat maltose, you have to break it up into two glucoses so that you can use this glucose molecule for energy. So remember a moment ago we looked at that area that's chemically different from, that could be very specific to a molecule, and that area we're going to call it the active site. This is the site of bonding to the substrate. This is the place that is perfectly engineered. And again, I say engineered, implying that there was some kind of forethought to it. It might have been randomly created, but in any case, you have this area that's chemically perfect to bond to the substrate. In this case, it's maltose. So step one, maltose binds to the enzyme at the active site. Step two, the conditions are perfect for maltose then to break up, and maltose breaks up, and you have two glucoses. Please notice, it's very important that you notice that the enzyme never really changed. It wasn't chemically altered. It's still there, ready to grab another maltose and start the same process over and over and over again, as long as there's maltoses and as long as nothing's breaking up the protein. So the enzyme is acting like the matchmaker. A matchmaker, if you remember us talking about it, a matchmaker takes and brings two people together and allows for a bond to form. That is what enzymes do. They're either going to create the bond or take the bond away or break up the bond, but it's the matchmaker never is actually involved in the bond. It's just he or she is just bringing the two subjects together, and that's exactly what the enzyme does. It brings two things together. In this case, of course, they're not humans, but they're two chemicals, or two, in this case, molecules and these molecules can either be bonded together or a molecule can be broken apart. But whatever happens, the enzyme creates an environment that allows for that reaction to go forward much more easily, because it actually literally brings them together or positions them in such a way that the reaction is likely to happen. So we have an enzyme here. This is the same enzyme, that catalase enzyme, and what you're looking at is a space filling model. So you see each of the atoms actually is relatively the size that it should be. So you have small hydrogens, and you have large carbons, and very large oxygens, and you can see how they form a shape. They form a shape that's very, very distinctive to to the molecule that is going to fit in that shape. So you have this molecule that's going to fit into the space that the enzyme has for it. It's going to fit into this space, and as it fits into this space really neatly, it's either going to be broken up, or there'll be another molecule that's going to be brought into the same space that allows for the two molecules to either bond, or this one molecule to be broken up into two. So if we look at the enzyme and we look at the reaction, this is a chart that you've seen over and over again in class. You've been tested on it. Everybody kept confusing the catalyzed or enzyme reaction line with the normal chemical reaction line. But you see, this line here represents the line of the chemical reaction as it would flow without a catalyst, without an enzyme. So when you're taking a look at this chart, you always have to remember you're looking at a couple things, and the two things actually make sense if you think about it. This is the uncatalyzed reaction. This is the line that happens without the enzyme. This is the reaction that happens with the catalyst. This happens with the enzyme. Now, if you notice, what's the difference between the blue line without the enzyme and with the enzyme? What's the big difference? So what's the difference between the two reactions, the ones without the enzyme or the catalyst, or the ones with the enzyme and or the catalyst, right? So the catalyzed reaction or the uncatalyzed reaction. The difference is the activation energy, the EA, the energy of activation. Really, that's what we're talking about here. If you can think about the idea of the enzyme bringing in the reactants, bringing them close together, or creating an environment where one reactant can be broken up more easily, then I hope you can see that you need less energy in order to activate the reaction, make the reaction go forward, to make the products. That's really important that you have to understand that, because that's how an enzyme makes the reaction go faster, just like you saw in the lab. Okay, so your lab experiment. Well, your catalase experiment had a pretty clear objective, and that objective was just simply to figure out or answer the question: does the enzyme catalase work better at a cool temperature, at a room temperature, or at warm temperature? Then the second question was, does the enzyme's concentration, in other words, does having 1 unit of enzyme versus having 2 units of enzyme, or versus having 3 units of enzyme, does that impact the amount of oxygen that's produced when the enzyme helps break H2O2 into water and oxygen? There were 3 trials for each temperature, because you had to see what was going to happen with one enzyme, one unit of enzyme, what was going to happen with 2 units of enzymes, and what was going to happen with 3 units of enzyme in each temperature. And of course you had to have a control group, so your control group was having just H2O2 in the vessel versus your experimental group, which had the H2O2 and the enzyme. Now, if you that took care of the control group and the experimental group. But now that comes the question, which one is the dependent variable and which variable is the dependent variable? Well, to decide which was the dependent and which was the independent, which one was dependent and which one was independent, you had to ask the question, which of these variables, the temperature, the concentration, or the amount of oxygen produced was dependent on the other. If you think about that, if you think about it just clearly, you know that the temperature is something we controlled, so that was independent. The concentration of enzyme, or how much enzyme we put in, was also independent because we controlled that. We put that in. Changing the temperature, for instance, did not change the concentration, and changing the concentration did not change the temperature. But the amount of oxygen gas produced was dependent. And it was dependent on what? It was dependent on the temperature or the concentration, because those are the two things that we changed. So now you know which one's the independent and the dependent. Remember, the independent would be graphed on the x and the dependent on the y. So here's the class data for the experiment. And remember, we were looking at units of oxygen is produced. And that was going to be, we were going to look at that as a relative measurement. So it was either going to be a 0, a 1, a 2, a 3, or a 4, depending on your observations and your gauge as to how much was produced over a given amount of time. So as we look at this, if we just have just hydrogen peroxide, everybody agreed that for the most part hydrogen peroxide by itself, under all the conditions, had virtually no oxygen being produced. So there were no bubbles. If there were a couple, there might have been some experimental error. Maybe someone had a bubble in there from pouring it in, or perhaps there was some residual enzyme from someone else's experiment. But the bubbles were virtually zero. By the way, those are two sources of errors that you might be able to discuss during your lab write-up. Now, when we take a look at what happened when you had 1 unit of enzyme, now you have that happening across 3 different temperatures. Temperature A, temperature B, and temperature C, right? The room temperature, the 0 degrees, and the warm. You see I think you'll notice that in the B, at the 0 degrees, it seems that people were getting more bubbles than in the warm. That seems unreasonable, but that's the units, that's the results they got. We'll talk more about what should have happened and what were some of the sources of error. Now, because of time, we had to have a group do 0 degrees, a group do room temperature, and a group do warm. Now, what that means is that when we're looking at relative measurements of either 1, 2, 3, or 4 units, Group A could have, or room temperature group could have been gauging it differently than group B, and that could have been gauging it differently than group C. So you could have different measurements, and that would confound the problem, confound the actual analysis of the results. However, within the one thing that we can look at, and it could hope that we could find some relative measurements that make sense, would be the change in concentration of enzyme. I say that because within the concentration of enzyme, where we had either 0, 1, 2, or 3 units, you'll notice that that was one group. One group made those relative measurements, so that we can take a look and we can have a little bit better confidence that the group that said that this was 0 and said that that 1 unit gave them a 2, and that 2 units gave them a 3, and that 3 units gave them a 4, that group made some really clear measurements of relative measurements that were consistent, where there was probably some inconsistency between the groups. There, more likely than not, was not much inconsistency within the group itself. And as you can see, all 3 of the groups, Group A, Group B, and Group C, each made pretty clear that it was pretty clear that definitely one group 0 enzyme had less bubbles than 1 enzyme, which had less bubbles than 2 enzyme, which had less bubbles than 3 enzymes, units of enzymes. So even though there was some inconsistencies between the temperatures, we can see that there was definitely some consistent results between concentrations of enzymes. And that would finish your analysis of error. Now, how are you going to graph this data? Well, you have 3 groups, and you have a lot of choices to make. You could start by trying to graph the average of each of the information. That would probably be the easiest thing, but may not give you the best results. You could graph each individual group's data separately, but on the same graph. How could you do that? Well, you'd have your x and y axis. We know your independent data is either going to be, you'll have 2 graphs, and I'll do the second one in a minute, but you'll have 2 graphs, and the first graph is probably going to be concentration of enzyme on the x. Now you're going to put it on the x because it is the independent variable, and that's either going to be a concentration of 1, 2, 3, or 4. Now on the y-axis, you're going to put the amount of oxygen produced in units, while on the x you have the concentration of enzyme in units. What's clear here, of course, is in the units of oxygen produced is going to be either 1, 2, 3, or 4. Now, if you graph the data and you say, well, of course, there's also 0, so I should add 0, 1, 2, 3, or 4. If you graph the data, then and you know what? I shouldn't put the 0 there, should I? I should put the 0 at the bottom, right? So here's 0, here's 1, here's 2, here's 3, here's 4. So if you're going to graph the data, you can say, okay, Group A, which was the room temperature group, could have a 1 here. Maybe they said it was 1 for 1 concentration 1. Maybe at 0 there was, excuse me. If we look at the data again, and we see that we have at zero concentration, there was 0 bubbles. That was your first and clear point. Then, after your initial control measurement, you saw that when you increased it to 1, you probably got a 1. When you increased it to 2, you probably had a 2. And when you increased it to 3, maybe you got when you increased it to 3, you may have gotten a kind of probably a little less than 4, so something like a 3 or 3.5. When you take the averages of well, I don't know if you can do the averages or not. So that could be what group 1, right? What group 1 came up with. Where, notice I'm going to change the color of the line, and I'm going to change the shape of the points, right? So that I can clearly distinguish between group 1 and group 2. So maybe group 2 still had, well, they had their initial point was 0 as well, but then they had at 1, maybe they measured it as 2, and maybe that at 1 unit, at 2 units of enzymes, they said it was kind of like a 3, and then at 3 units it was a 4. So maybe they're kind of graphing it more like that. And if we look at the third group, which might be, I don't know, cold temperature, they still get the 0. They still get the 0 there. 0 concentration gets you 0 bubbles of oxygen. And maybe they match up here, but maybe they get 2 and they still think it's more like a 2.5. And maybe the 3 is more like a 2.5 or 2. And their graph ends up looking more like this. So then you have 3 groups. You have group 3. You have this line, which represents, maybe it represents 0 degrees Celsius, and maybe this line represents a warm, and maybe this line represents cold. So there you have a graph constructed really quickly, sketched out how you would graph this data using groups 1, 2, and 3, or ABC, however you want to call it, without having to take an average. Now, if you took the average of all the 3 different groups, which could help because maybe that reduces error, you don't get as much detail as far as maybe one group did better than the others. But if you take the average, you'd only have one line to graph because each of the points would have been averaged out rather than having the 3 separate lines that you see here. Okay, well, I hope this helped you. We'll send you another video with how to do this using Excel, and I hope this helps you with your lab write-up and your understanding of the Enzyme Lab.